Hello, everybody. I'm Dr. Amir Abbas, and I welcome all of you, whether you are watching this session live or you are watching the recording of this session. Today, we have a very important session and panel discussion on current practices in healthcare research and analytics. For this purpose, we have three guests apart from myself as speakers. Our first guest is Rabia Khurshid. She grew up in Bhawalpur, Pakistan, and did her master's in virology and immunology from NUST Islamabad, and taught at Islamic University, Bhawalpur Institute of Noor International University, Lahore. She is currently doing her Ph.D. in cancer biology at the University of Miami Miller School of Medicine. Her research involves systems, drug discovery, and is currently working on projects in both cancer and infectious diseases. So first of all, we'll welcome Rabia to the stage. Thank you, Rabia, for giving us time today.
Thank you, Amir, for inviting me for this event.
Okay. So, and we are especially thankful to Rabia because she's not feeling well. We hope and pray for her that she feels well. Okay. Our second.
Thank you.
Okay. So, our second guest is Salman. He's a Fulbright scholar, and he's pursuing his doctorate in applied economics from Claremont Graduate University. He's interested in solving social problems and investigating the causal links of social behaviors using economic theory and data. These social problems include a wide array of topics from public health, policy, and to very complex problems like the formation of cultures and mass behaviors. He had worked with the team from Information Technology University on the immunization project, which is known as Har Zindagi, which has rolled out in 6 districts of Punjab. Moreover, he also worked with Pakistan Poverty Alleviation Fund to investigate the behavior change of voluntary organizations due to self-reporting of their activities. So with this, I will also like to welcome Salman to the stage. Salman, thanks a lot for giving us time on this weekend morning. How are you?
Thank you for inviting me over.
Okay. And our last distinguished guest is Osama Shiraz Shah. He's a Ph.D. scholar from University of Pittsburgh, currently doing research on breast cancer. His thesis project focuses on characterization of molecular, cellular, and spatial feature of mixed ductal and lobular carcinoma, and identification of which of these features are unique or shared when compared to IDC and ILC subtypes. His areas of interest are bioinformatics, breast cancer, and epigenetics. So let me bring Osama to the screen as well. Thanks, Osama, for joining us today on this weekend morning.
Good morning. Thanks, Amir. Happy to be here.
Okay. So without any further delay, I will like to invite our first speaker, Osama, and I think he said that he wants to share his screen. So go ahead, Osama. You'll be able to share your screen as well, and the stage is yours.
Thanks, Amir. Okay, I will try to share my screen. Is it being shown?
Just a minute. I think not yet. Oh, yeah, it is here. Yeah.
Great. All right. Great. Okay. All right. Thank you, Ahmed, for inviting me. And today I'll just introduce this this area of genomics research that is trending right now. And I'll just briefly introduce some of the tools that we use in genomic research, specifically focused on breast cancer. But I will try to make it generalizable as much as I can, and how we do use these tools to answer some of the healthcare questions that are pertinent to improve disease management. So with that, I'll just start. So this is the outline. First, I will briefly introduce what the genome is, how does the genome work, what causes the genetic diseases, how to study complex genetic diseases, and what are some of the genomic approaches that we use to study these, and how does genomics play into the cancer healthcare. And I will end with one case study, which will highlight some of the findings from our work, which are focused on breast cancer genomics and precision medicine. So first, the question of what is the genome? So genome is just a fancy word for all your DNA. So if you look into a cell, you have the nucleus, which packs in the chromosomes. And if you open up a chromosome, if you uncondense it, you see these long strands, which if you look under a high resolution microscope, you can see that they are double stranded, and basically the way they are double stranded is that there are bases A, T, C, G, which form base pairs on both sides of strands, and making this double helix structure. There are 3.2 billion bases in human genome as a whole. So the DNA does not stay like in uncondensed form. It's actually packed into these superstructures or higher structures called chromatids and chromosomes. So there are 23 chromosome pairs in human. The next question is, how does the genome work? So you might have heard about the central dogma. This idea that how the genomic code becomes function is shown here. So if you look at the left, you have the double helix structure of the DNA. And let me get a pointer. So, sorry. So this is a double helix structure. The first thing that DNA does is it replicates itself so that it can preserve itself. That's very important because everything, the raw information, all of that is in the DNA. So it will replicate itself to preserve itself. The other thing that happens to DNA is the transcription process, where you get RNA made from the DNA. So if you think in layman terms, DNA is just the raw instruction that gets processed or that gets transcribed into RNA, which is the process instruction. And finally, this process instruction or RNA is translated into the functional units of everything like which happens in the body. So these are called proteins. So not all of the genome gets transcribed. There are certain regions of the genome which are called the basic units of instruction or genes that get transcribed, translated, and form functional units called proteins. In total, there are around 21,000 genes in human, and they define how we look, what our eye color would look like, what our hair texture would look like, and other things. So coming to this question of like what causes the genetic diseases. So if you think about the genetic diseases, basically most of these diseases are associated with changes in DNA, which we call mutations. I've put here 3 types of genetic diseases that we'll look into. The first category is of diseases like cystic fibrosis, sickle cell disease, which we have heard about. And these happen due to mutations or changes in DNA for one gene. So these are called single gene diseases. Then you have changes happening at a larger scale at the chromosomal level. And one of the examples of a disease in which 21st chromosome gets an extra copy is Down syndrome. Then you have more complicated diseases like cancers and cardiovascular diseases. And these happen due to various genetic factors, could be multiple genes, chromosomes, and other things. And in addition to these genetic factors, you also have environmental factors playing into role, like smoking or UV radiation. So traditionally, these single gene or more simpler diseases have been studied using genetics. But the study of these complex diseases where you have multiple genes interacting with each other and then the interactions with the environment, you need more sophisticated tools. And that's what genomics is. It's a study of the genome, including the interaction of the genes with each other and the environment. So genomics, again, is a very broad field, but some of the key things that are being used in genomics, these include genome sequencing, genome editing, bioinformatics, and their combination. So when we look at genome sequencing, there are many approaches, but some of the key approaches that define genomic sequencing are DNA sequencing, which quantify basically the changes in DNA, which we talked about. So these are the mutations. And then we have the RNA sequencing, where you quantify the individual fragments of RNA, which when you quantify, you get information on the changes in the gene expression. And then you have genome editing approaches like CRISPR, which we have all heard of, and shRNA. And these are methods that people are using in genomic research to validate different things in disease models like cell lines and mice. And then you have the bioinformatic approaches. So given the wealth of data that is being generated given that the genome is so complex, there's so much information there, you need sophisticated computational methods to understand what the data that you have produced means. So some of the bioinformatic methods that are commonly used are sequence alignment. So when you sequence a genome, you get a lot of information, basically in terms of sequences. You get a lot of sequences, and now you are interested in like which part of the genome is this sequence coming from. That's where you use sequence alignment. Then you have this other method which is commonly used, is called differential expression. And basically what this does is when you do RNA sequencing, let's say of a cancer sample, and you want to compare that with a normal sample, you do RNA sequencing on both, and then you try to see the differences in the gene expression. So that's called differential expression. And then finally, I mentioned one more tool here, which is mutation calling. And what this basically means is comparing the sequences just to see what is being changed, which part of the DNA is being changed. So having introduced the genomic approaches, now I'll just give a few case studies. We can see like how these apply to different aspects of the healthcare before I move on to the case studies. So if you think about the healthcare, there are key aspects of healthcare and disease management which are being aided by genomics. So let's think about diagnosis. So if you think about diagnosis, we can think about identification, that genomics can help in identification of disease biomarkers. When you think about the treatment aspect, we can think about how genomics can help identify drug targets. When we think about the prognostic or the prognosis part of the healthcare, we can use genomics to do prediction of treatment response and disease outcome. So I will now introduce like a case study, like our workflow of a case study, how we use genomics in cancer healthcare. So let's say you have a cohort of patients. You have various cancer patients. In this case, let's say breast cancer patients. So what you do is you collect RNA and DNA from them. That's the first step. The second is you do sequencing. Let's say we do RNA and DNA sequencing, and then you get some raw output from the machine. And this itself is not reliable. So you use bioinformatics and data analysis to understand what this means. So overall, this is what the genomic pipeline looks like. The analysis output would look like something like this. So if you did DNA sequencing, you would get the changes in DNA, as we discussed before, or the mutations, right? And these are just the sequences that you get, right? And these have been aligned to a particular loci in the genome. So this is, in this case, it's chromosome 17, some specific location in chromosome 17, and these are the sequences that we got from the DNA sequencing machine, and these are now aligned to that particular loci. So you have the tumor sequences or the cancer, the cancer sequences, and you have the sequences that you got from the normal samples for a comparison. And now you can see that there's this particular change in a particular nucleotide in this location. So initially in the normal sample it was A, and it's being changed to G. So as we discussed, these are what we call mutations or DNA changes. On the other hand, you can also do RNA sequencing, and then you can get information on the gene expression changes. And here, this plot basically is showing many genes on the rows, and the samples are shown on the y-axis. And the samples have been divided into the tumor and the normal samples so that you can see the gene expression patterns. So this area which is highlighted here is basically showing tumor expressed genes. The color basically red means the highly expressed genes, and the low means the ones which are expressed in lower level. So overall, from this analysis output, we get we get output of mutations in the tumors that were profiled or analyzed using a genomic profile, genomic pipeline, and then you get the genes that were expressed in the tumors. So both of these informations could be used to define potential biomarkers and drug targets. So once you find these potential putative biomarkers and drug targets, you first validate them in disease models. So in this case, we are working with breast cancer cohort. So we will use breast cancer mouse models or breast cancer cell models to validate these, and then these could then be used to improve disease diagnosis and treatment in clinic. I will show one more case study here, or like a workflow here, just to highlight another aspect, which is the prognosis one. So let's say you have a drug in the market which is being used to treat cancer patients. And the observation that you see after treatment is that there were some patients that responded to this drug that you have developed, but there were some patients which did not respond. So we'll call them non-responders, and the ones that responded as responders. And the question here is what are the genomic differences between these responders and non-responders? What is so special about these the genomes of these responders that they respond, and what is so different about these non-responders that they're not responding? So again, we'll use a genomic pipeline that we just discussed. And here again, we get the DNA changes and the gene expression changes. So instead of thinking of the tumor and normal comparison that I showed before, now you can think of this as a responder and non-responder comparison. So again, we can see the mutations that are present in the non-responders, or you can see what are the things that are associated with response. At the mutation level, the DNA change level. You can also see this in terms of the expression change for the genes. And both of these together would help us define potential prognostic markers or predictive markers, so that we can find which individuals with a particular disease would respond to a treatment and which would not. And similarly, we'll validate these in disease models before we use them in clinic to improve disease diagnosis. So finally, we'll share some of our own work from my lab. And here I will just introduce this concept of precision medicine with this. So in this study, which are cited in the bottom, we basically develop a diagnostic and prognostic kit using the sequencing or NGS or genomic methods. So first step of this was to basically go through these genomic studies on breast cancer and identify the mutations that were associated with advanced breast cancer. And then we made this kit. It's called MAMA-seq, which is for both prognosis and diagnosis. So for the diagnostic part, I'll just share the next panel. So this is a panel of results showing how the MAMA-seq was used to detect mutations. So again, we had a cohort of samples from where we collected the DNA, and then we applied the MAMA-seq genomic pipeline. From this, first thing that we get is that are the mutations in different genes. So this panel, what it's showing is that each column is basically sample, individual sample. So everything in the shown on the columns is the samples, and each row represents a gene. And different colors in the plot, they represent different mutations or different changes in the DNA that are happening. And what this is highlighting is that although all of these are breast cancer patients, they have different genomic mutations, and based on that, you can see which of these could be targeted in a different way or treated in a different way. So people who work on breast cancer, they know that this ERBB2 gene is a very important breast cancer gene. Is associated with poor survival and all that, all the problems of disease progression. So some of these patients have this mutation enriched. On the other hand, many others don't. So they have different mutations that were enriched in different genes. So based on this information, you can define druggable mutations, right? And there are many databases that are being developed right now, two of which I have put here, PMKB and OncoKB. These are so-called precision medicine databases where they have information on the genes and the mutations that happen in them, and if they're druggable or not. That means if there are drugs available in the market or not for them, or in different phases of the clinical trials. So from this set of information that we got, which is the mutations, we could see which mutations would be druggable. So these two panels just show overall that MAMA-seq kit that we developed was used to diagnose different mutations and then select the drugs that could be targeted for each individual. So this is the concept of like precision medicine, finding specific targets in individuals and then targeting them in that way. Second part of this study was basically the prognosis. So here I will show the example of how MAMA-seq was used for disease prognostication. So there was one individual that had terminal disease, and they were being treated with different drugs. And at different time points during this clinical care, we took DNA samples. And using these DNA samples, again, we did the mutation. We found the mutations that were getting enriched. And specifically, I will talk about a few mutations that were associated with treatment response. So in the bottom plot, what this is showing is the timeline. So the time is in months. So from left, you have the first month till the 27th month. And in the bottom color bars are showing different drugs that were given. And these symbols here are showing basically at what time points the samples were collected from this particular patient. So what this plot is showing is that initially the patient was being treated with hormonal therapies. So these are the go-to or standard therapies for breast cancer patients. Eventually, what happened was you can see that these points are showing that if the patient is responding or not in terms of mutational burden. So if the mutational burden is high, that would mean that the patient is not responding. So initially, you can see the mutation burden is low. So the patient was responding to these therapies. You can see some blips, but overall, everything looks good until this 10th month. What happened was they actually got mutation in this ESR1 gene, which is responsible for response to hormonal therapies. And this particular mutation made the patient resistant. And then there was a change of therapy to mostly chemotherapy and other therapies. And then you can see the patient started responding again, and the mutation burden went down. But eventually, the patient got more advanced disease, and it spread throughout the person's body. And that's why overall the patient stopped responding and they passed away. But this just shows a case study like how DNA sequencing could be used to identify the mutations which are responsible for a particular drug resistance, and how these mutational burdens change over time as the patient is treated. So overall, this is the model that is now being made feasible due to genomics, and it could be used in healthcare. So I'll end with a summary of things, which is that mutations in genome can result in diseases. Complex diseases are a result of multiple mutations and various environmental factors. Genomics uses combination of genome sequencing, genome editing, and bioinformatics approaches to study these complex diseases. Genomics can improve various aspects of healthcare, like diagnosis, prognosis, and treatment. And finally, with the last case study, what I highlight is that genomics is paving way for the precision medicine-inspired disease management. There are many genomic resources available right now, so you can there's like too many of them, but some of the key ones that we often use in our cancer research, genomic research, are listed here. So I'll end with that. Thanks for listening.
Right. So Osama, this is an impressive and comprehensive talk. I wish we had more time. Personally, I have a lot of questions, but in the interest of time, we'll move forward. And for those of you who are watching it live, you can put your questions in the comments. We will generate a bit of discussion at the end, and we'll take questions from the comments as well. Without any further delay, I will move to our next speaker. She's Rabia Khurshid, and she'll be talking about computational methods for drug discovery. So over to Rabia Khurshid.
Hello, everyone. Hope you guys are doing great. I'm going to start sharing my screen now. Oh, wait. I have some.
That's okay. You can take
preferences.
That's okay. In the meantime, as Rabia is sharing your screen, it's a request to all of you to Oh, we dropped Rabia. She will just come back. And I will take this opportunity, as we have right now, to request everybody to put your questions in the comments. And at the same time, I will also request to those who are live to share this live stream, because if more people are watching this and they ask questions which are informative to all of us, we'll be able to generate very useful discussion. So we have Rabia back over here. Yeah, that's okay. Rabia, we lost
try
you.
again.
Yeah, you can.
Because something is.
You will be able to see a share screen option.
Yeah, I'm put entire screen, but it. Oh, okay. I had to click on the entire screen.
Yeah, here.
Can you guys see my screen now?
Yeah, I have your screen over here, and when you're ready, let me know. Yeah. Okay. You should be able to.
Okay, so I will be talking about system drug discovery approaches. We use systems drug discovery because the biological system is very complex. It's not like we can just target one thing. Of course, there will be tons of things that can go wrong. So you have to keep in consideration every other pathway that intersects with what you're targeting. So there are several approaches that you can take in systems drug discovery. You can take target-based approach, which is the development of a drug that selectively modulates the effect of the disease-associated gene and gene product without affecting other genes or molecular mechanisms in the organism. Or you can do drug repurposing, which involves identifying new therapeutic uses for old or existing available drugs. And you can also do drug combination, which is the best among these three, which involves when you use two or more drugs to produce an effect that is greater if either of the drug was taken alone. And this is called as the drug synergy. So over here, can you guys see my cursor? I think I need a laser.
Yeah, we can see your cursor.
Oh, okay, thank you. Yeah. So you can, for an example, you can get the disease data. Here, I'm talking about the TCGA data, which is the cancer data. You can take that data, and then you can take the drug data, and then combine them to get a drug that is effective against that disease or cancer in this case. Or you can enter that data into a platform like SynergySeq, and then it can combine the data for different drugs and come up with a combination of drugs that will have a 2x effect against the disease as compared to the initial drug that it came up with, our study came up with. So if you want to do drug discovery, what will you need? You will need high-speed computing servers and storage. You don't have to have a lot of computer knowledge yourself, but if you have a computer geek in your lab or in your at your workplace, that will work too. And then you will need a chemist, because then because once you have a drug, they have to look at the structure of the drug and come up with whatever changes you need to make to that drug. And then some softwares, they need licenses. If you are in a university, part of like a university or any academic institute, then you might get some academic licenses from some of the softwares, but some still need you to buy a license from them. And then you will also need to have a really nice PI. If you're a student, I think this is very important. And I'm glad my PI is the best. And then you also will need VPN for remote connection to your workstation, because it's the COVID times and I'm working from home, and I'm glad that I have that. Moving on, where do you start? So you will start with databases. Once you have identified a disease that you're interested in working on. So there are, for example, if you want to work on HCV, which is hepatitis C virus, then you're gonna go to PDB, and then you're gonna look for all the data that is related to HCV, and And PDB has, it's an open repository, and it has all the resolved crystal and co-crystal data for these proteins and the ligands, or you can say markers and the drugs. Then you have another database that is called Campbell, and Campbell is manually maintained database for all of the bioactive drugs against different diseases. And you can search Campbell by drawing the structure of the drug that you think you are interested in testing against your target, or you can just have, even if you have like a part of a drug structure, you can just search on the basis of that, and it will come up with all of these different structures that are similar to your drug. And you can also search by disease or by the drug name, and you can download all of this data in bulk, the bioactivity data, and then you can use that to assess which drug is the best for your target. The second thing you will need would be some softwares. So once you have data from PDB and Campbell, you will need to visualize your proteins and run docking studies in MDs. So we use Schrodinger Maestro. This for using this, you will need a license. And our lab has an academic, not an academic actually. We have like a lab license for this. And if you just want to just look at a project, you can just download the academic version and you'll be able to just look at a project. But if you want to run docking and MDs, then you will need to get the license. And there are scores of things you can do in Maestro. You can do visualization, you can prepare the proteins, you can do ligand prep, you can do MD simulations, docking, and then you can also do lead identification and optimization. You can compute physiochemical properties and enumeration of the compounds. You can also do data visualization and analysis. But what you can't do in Maestro is the protein-protein docking, which is very important if you are looking at complex drugs. So for protein-protein docking, we have other platforms. We have Haddock, which is like a server, a server where you can submit your data, and they will use their computing to do the docking for you, and then you can download the results. You also have Rosetta. You can download that I think it's for free. You can download it, but if you're good with command prompt, then you can install it on the server and then use it through the command prompt. But if you like using the user interface, then you will need to download the PyRosetta, which is interactive and easier to use. Given that, but you can only download these if you have up-to-date operating systems. If you have like older Windows, then these probably won't work. And so after you have your drug, you have narrowed down on your target and your drug, and you have you're looking at your molecule. And sometimes you need to make some changes to your drug, and for that you will need some other softwares. And what we use is what you could use. There are others too, but what we find really helpful is the Marvin from ChemExon. You can get an academic license for that, and it's free. And you can draw structures and then search them on Campbell, and then save those structure files so you can go back and make changes to your drug later. And also you can, it's also really helpful if you want to download high resolution pictures of your structures to use in the publications. Another really interesting and important tool that we use is the Pipeline Pilot. The Pipeline Pilot lets you do data processing and and analysis of the data. And it has a lot of different, if you look on this side, on the left of this panel, it shows all these topics, and there are tons of components that you can add to your pipeline that you can build depending on what analysis you want to do. So here it's the example. What you see here is the example of the enumeration of these properties for some compounds that I did, that I built. So first, you can see here it's reading the file, and then I added a component that changes the molecules to structures from SMILES. SMILES is like is how you write a structure so it's readable by computers. So and then it changes it. I don't know if you guys are familiar with Canonical, but it's also important when you're working with SMILES. And then I added all these components: molecular weight, AP log, log D, because I wanted to calculate these. And then you can either view these the results of these or write it to a file. So this Python pilot, you can do scores of different things, from data wrangling to machine learning. There are machine learning tool and components here that you can add to it. Moving on, you will also need access to the servers, so that because it's really important for you to store the data as you work, because if you lose if your computer crashes, your data is gone. So we use FileZilla to store our data on the servers. It's really easy to use, and it's free to download. So over here, I have an approach overview, a simple approach of how you would go about docking some compounds against your target protein. You first have to go to the scientific literature and identify what you want as your target, and then you're going to download data from PDB, the protein data bank that I talked about. And then after you have the choice, the target of choice, then you're going to go to Chembl and download all of the compounds that are found to be active against this target. And then you're going to do chemotyping of the compounds using ChemExon or the Marvin tool to see, to categorize all those compounds, because there are like thousands of compounds, and you can't do anything manually. You have to have some software for that. And then you're going to do ligand docking in Maestro, Schrodinger. And here it says release 2019-1, but now the latest release is 2024. That has some added features. And then you're going to have the docking results, and then you'll do the analysis and then plan your next experiments. And we do the analysis in Spotfire. It's a really good tool for data analysis. And here's a machine learning approach example. This is really, really interesting. So after you get the actives from Chembl, you can go to this website called Dude, a database of useful decoys. And what they do for you is that you submit your actives to them, and they give you a list of inactives, which are called the decoys, which are the compounds that are not active against your compound, your protein or target of interest. And then you'll go to Pipeline Pilot and use the naive-based machine learning classifier to train a model, and the model will learn to identify the actives and to exclude all the inactives on the basis of these two decoys. And then you can go to the eMolecules database, which has millions of commercially available compounds, and run those through this model, and it's going to give you initial predictions, which will be in thousands. And then you can filter the top 10 or 5% of those, and then you'll have several thousand potentially active molecules, and then you can do further analysis on those and look at the structures and have a chemist look at the structures and see if those are those compounds can be made in the lab, if they're easier to synthesize. And then once you have those molecules, you can then either synthesize those or buy those because they are commercially available, and then test them in cell-based assays and so on. So this is all from me for now. Thank you so much everybody for listening.
Thanks a lot, Rabia. Once again, it was a very enlightening and though introductory presentation, because we would like to know a lot of the details related to these areas. And I'm sure that we will talk a bit more about these areas in the discussion session. And without any further delay, I'm going to move to our next speaker, Salman, who is going to talk about how to establish causality with the help of a cross-sectional study, because he's an economist. So he'll be using and telling us how to use econometric methods to infer causality from a cross-sectional study. Over to you, Salman. Sorry, over to you, Salman. I'm sorry.
Hi. So thanks, Amir. So I will start with the very basic question that why this causal relationships are important. So if you look at so if you go over the internet and download the consumption of chocolates data, and then download how many Nobel Prize winners each country have, and run a regression, and you would see that the chocolate would be a significant predictor of winning a Nobel Prize for a country. And do you think that's a significant predictor? Obviously not. The chocolate cannot predict that if a country is going to get a Nobel Prize. So what's the problem here? The problem here is that this link is not causal, and most of the time what happens is that there is another factor that's driving both our dependent and independent variables. So in our case, there might be some variable that's affecting chocolates, and that's also affecting the Nobel Prize winners, and we don't know about that. So if that's the case, then we might get significant results with these types of relationship, and we call them spurious relationships. So the problem with establishing causal relationship is very complex. So we need to find. So let me give you an example. That let's say I have a headache and I have to take a medicine. So if I take a medicine, then maybe I'll get better, but I won't know that what would have happened if I would not have taken that medicine. On the other hand, if I did not take that medicine, then I would not know what would have happened if I have taken that medicine. And the comparison of these two scenarios give us true causal relationship. So the problem here, the scenario in which I do some action and observe an outcome is we call it factual, and the other one where I did not do the action and observed that outcome, that's called counterfactual. So the problem in establishing causality is that we never had a counterfactual, and even today, all the causal relationships do not have counterfactuals, do not have actual counterfactuals. So all we do is that to create that counterfactual as effectively as possible. So how do we establish causality in medicines? And you might have, you all would be very, very familiar with that they do randomized controlled trial in which they have a random group of people they give medicine to that group, they call that treatment, and there's a control variable, and the placebo would be given to that control group, and then they would compare these groups and say something about causality. But remember, again, it would not be a true causal relationship. Why? Because we would not know the outcomes of treated people if they would not have taken medicine. We are just trying to say that the control group would perform similar with if they have taken medicine. So, and that's a very bold claim to make, because in reality we don't know that. So that's why we randomized, and with a complex mathematics we try to prove that this control group is actually approaches to the counterfactual. And in medicine we double blind the experiment. So the double blind is that both the participants and the researcher doesn't know that which group are you in. So there's a whole lot of literature on the participant not knowing if he was in the control group or treatment group, because in medicine there is a placebo effects. But what about this researcher not knowing that you are in the patient he is administering the drug is in control group or treatment group? That seems trivial, but it's not, because in US in 1960s there was a horse show in Kansas, and what happens there is that one horse has a ability to do the maths. So people would come and ask questions like 2 plus 4, 3 minus 2, and the horse would kick his leg to the ground that many times, and every time the horse was correct, and everyone was amazed. Then these group of researchers went there and tried to find out what's happening there, and they talked to the owner of that horse, and he said that I just started training my horse. Like I would ask some questions, these mathematical questions from him, and then if he gives the correct answer I will give him food, otherwise I won't give him food. And that's how he learned the mathematics. But for the researchers it wasn't possible that a horse could do this basic algebra. So what they did that, let's try. But this time they asked the owner to not be in front of that horse, and then the horse could not guess any answers. So what was happening there? The horse did not learn to do the basic maths. He just learned to read the facial expressions of his owner, and that horses can do. So you see that in establishing causality is how complicated is that. When you see this setting, you would say that by just conditioning the horse you can teach them basic maths, but no, you can teach them how to read facial expressions and not maths. So that's the intuition behind double blinding the experiments. But in a experimental control setting, it's very easy, not very easy, but it's easier to set up a randomized control trial. You have a group of people, you randomly divide them into groups, and do your experiment. But in social settings where you have health policies implemented and other kinds of policies, it gets very difficult. Why? Because you cannot control people's life. So if there is a policy, like in our example, we have a redesigned immunization card, and we give them to our treatment group, but we cannot control their lives. So we don't know what's happening in their life, what's causing them to change their behavior. Is it that card, or is it some other thing? So when we are in a social setting, there are lots of variables that can affect and that can confound our causality. So, and then these randomized control trials are very expensive when you are in social policy settings. So the solution to this is to use observational data. So the first method that we can use to establish causality, it's not more of a causality, but to control for all the factors that we cannot see. That's fixed effect models. So if you have data over time periods, the same people with for maybe 6-7 periods of time, then by using these fixed effect models, you can distinguish what's observable and what's not observable. So for example, if I have a data for a class of students, then the ability, we all know that it's not unobservable. It might be observable, but you cannot quantify it. So but with using these panel methods, fixed effect models, you can remove that part of ability from the analysis so that you can focus on your treatment. The very powerful method that used to establish causality is the mixture of difference in difference and fixed effects model. So what difference in difference does is, again, it's the difference in difference started from epidemiology. John Snow, I guess in 1800s, have a problem in London that there was a cholera outbreak, and his hypothesis was that this disease is waterborne, but most of the epidemiologists at that time believed that it was airborne. And then one water company randomly, as per their policy, started cleaning their water, and this was a very good difference in difference model for John Snow. And without knowing that this is a difference in difference model, he used that variation because of this clean water to find the causal relationship of polluted water on the cholera outbreak. And what happens in this difference in differences model, that you have a treatment and control group, and these treatment and control group might be very different. So what you do is you first of all difference it to find what's that difference, that how these treatment and control are different before the treatment. And after the treatment, you again calculate that difference, and if that difference has increased or decreased, that would tell you what is the actual treatment effect. But again, we don't have counterfactual in that. We are just trying to say, if you think about this design, we are just trying to say that control group would behave similar to the treatment group. So if there was a difference of 2 points before the treatment, the difference after the treatment should also be 2 points. And we don't know that. That's our assumption. But based on that assumption, we use difference in difference and find the causality. Other than that, there are two new methods that's been using. So in difference in difference, there are we have this assumption, and most of the time people can argue that this assumption is violated because you have to establish that treatment and control were moving together before the treatment period. And that makes your claim that after the treatment it should have moved together a power. So but most of the time when you look at the data, treatment and control are not moving together even before treatment. So what happens is that we have a method synthetic control, and what synthetic control does is that it takes many control groups and try to weight it in such a way that it has a exact parallel trends before the treatment. So it's not an actual control, it's a synthetic one. So it comes from a data, but it has a lot of math behind it. So it's not real, but it can give you a proper control to compare with. Then most of the cases we have a cutoff points at which that policy is implemented. So, for example, in US, you can buy alcohol after you turn 21. So, if you want to find the relationship of alcohol on some factor, you can do it. But again, the people who were at before 21, like 18, 17, are completely different people who are much older. But the regression discontinuity design, what it does is that it just takes people who have just turned 21 and who are about to turn 21. These people should be the same, and then based on that, we can compare treatment and control to see what's the causal relationship of alcohol. Yeah, so these methods have a lot of maths and statistics behind it, but if anyone want to use it, I'm happy to take comments, and they can write me, and I would happy to answer the questions. Thanks.
Okay, thanks a lot, Salman. And like other speakers, I'm sure that this also is a huge field and it has a lot of details. And this was just an introductory talk of introducing, just like taking names of different methods which you can use. And though in our agenda, which was communicated, there was a next session on which I had to speak on machine learning, but I think I will just make a change in the plan right from now and let not the audience get overwhelmed with the information. And I would like to just shift to the question, answers, and discussion session right now. And because what I think that the audience of this channel perhaps wants to listen more from the visiting speakers rather than myself. I can speak separately on machine learning some other time. So there are many questions right now, not many questions in the comments, but a few questions. And I will start with a question which I have from the speakers. And this question is to Osama. You talked about your interest is in bioinformatics and you're doing a lot of research in that. So if somebody wants to come in and take bioinformatics as a career, right? What would you suggest? So my question is that, like, if somebody wants to adopt it as a career, what do you suggest to those people? Or if somebody like me who cannot, because he's a mid-career person, cannot shift career but wants to do research in this area, what type of skills would you like to me to learn at this point in time?
Yeah, I think that's an important question, and it's so compared to like other approaches that we use in genomics or in other, I guess, STEM fields, which are most related to like experiments. Those might be more challenging to learn, especially like in countries like Pakistan, because many of these things are very expensive. Even if you take like basic things like learning how to do sequencing, these things would be challenging due to the, I guess, the financial constraints that you have in countries like Pakistan and others. But learning like bioinformatics and all these, I think is much easier. Personally, for me, I started into this area during my undergrad, and it all started by just learning how to do coding. So it was just basic coding, and coding, like, I think we all have done at some point in some ways, or it's part of some of our curriculums as well, in science at least. And but it all starts like that. So just understanding how these bioinformatic methods use coding could be one of the basic ways to learn. And I think if you just go online, you can see there's so many resources available. Even if many places don't offer courses for bioinformatics, you can always go to, I guess, I personally also learn a lot from edX, websites like edX, Coursera. People have made a lot of GitHub pages where you can just see how people do a particular solve a particular problem and do a particular task. But yeah, I would just say start with the basics, which is just learning how to code. It could be just as basic as learning how to code in Python or R. And I think that would be the first step. And then you can eventually, if you want to go to genomics, you can start taking more of these courses from websites like edX and Coursera, where they have proper like syllabuses to like introduce genomics, starting from the basics. But you need to have like some understanding of how to code and do statistics.
Right.
I wanted to add some.
Yeah,
I'm sorry,
sure. Go ahead, Rabia.
There was a question in the comments I just read that what is the use of Python or coding in bioinformatics? So I would say that the simplest use would be to analyze the data, and then you can also there are some softwares that you need some code to in order to communicate with those softwares. How you give them your data, your specific data, depends on if you could write that code, or at least identify that code and find it online and use it. So what Osama said, just learn to code a little bit. And then also you can, I think really important use of code would be if you can get good at it, you can build tools for yourself so you don't have to buy expensive softwares. And then just so it would free you from anything that can stop you from learning bioinformatics or computational biology.
Right, and that is an excellent point, and which I was about to come to you, Rabia, for my next question, which is, and I want you to talk a bit in detail related to that, specifically to your area. But before we do that, we have a very relevant. I just want to take this question first because it is very much relevant to the point which we are talking about. It's Laura Mazur from an employee from Al Khan University, is our colleague, and she's thanks for today's session. I'm interested in knowing the career prospects in the field of bioinformatics and precision medicine for individuals in Pakistan. And the reason I ask this question that many people from there are some mind blocks. People do not know what bioinformatics is. People are not well aware, whether they will be taking some risk in, because it's a new field. So would you like to add something to this point, Rabia or Osama?
I'd say that so I did my master's from NUST, and I took courses in bioinformatics and molecular phylogenetics, and I thought that was the most advanced courses. Those were the two most advanced courses that I took, and I was learning something new. And I think right now, because that was like in 2012, and I think right now science in Pakistan has advanced a lot because there's now this medical university that is connected with NUST, and now the PhDs can teach at that medical university and not just the doctors. So so also because there is so much data out there and people don't know how to actually analyze that data, it's really important to have people on board who know how to read that data and analyze that and come to better conclusions than by just manually trying to figure out the answers. And I think it's gonna also, because in the field of precision medicine, I think that's what the future of medicine is. I mean, I don't think it is the future of medicine. So you need to have bioinformatics skills, computational biology skills to do that, to get good at that.
Okay, and Osama, would you like to add something to this point? And before you go ahead, let me tell you one thing. My computer is running out of charge, so you continue that, and I'm going to go off screen for just a few seconds to just put my computer on charge. Over to Osama for a few minutes.
Good, yeah, I think I agree with what Rabia said, and yeah, it's not that easy. I mean, in our undergrad level, like when I was doing my undergrad, I wanted to like try out and do new things in terms of research, but it's not that easy. Many of the research areas in Pakistan are still limited, and the things that we, I guess, some of the things that we talked right now, I think many of these things would take some time to even like become, I guess, more trending in Pakistan. But I think, the best part is that most of these things, like you said, about PDB, protein databases, a lot of the other data sets that Salman uses, and like the cancer data set, they are already available. So I think that's the best part. It's just about how, like, understanding how to use these. And as I said, I think many of these courses that you can take, the best part is they start off with very basic things. And you can just start off from there and learn how to do at least one thing to start with one data. And then you can like go on use more and more things once you are aware of the I think the pipeline in genomics has been like this. So you do the data generation. That's the first part, right? And you have some specific question, or maybe not. You just want to see like what the genome looks like in a disease patient, or maybe not in a disease patient, or in a different organ, let's say. But eventually what you do is you get the data, right? Now the first thing is what the data is, and then based on that type of data, you can just at least start with one thing and then see like how you can analyze that data. That will be the first thing. So once you become an expert, I guess, in terms of using these tools that are available, I think most of these tools are free. R is free, Python is free. Many of the tools that at least we use in genomics are just free. One of the websites that I mentioned, where you don't even need to like have many of these websites. So like CBioPortal is one that we use in for cancer biology a lot. It's a website where you can access data and just do the basic analyses. So many of these are already there. So I think using a combination of these, just seeing what the data is and doing basic analysis using these available tools, and then also learning yourself how you can do more comprehensive analysis using Python, R, or whatever you are interested in. And I think it's always a challenge to learn coding, but I think many of these websites, personally, when I use them, I feel like they do introduce from very basics. So I think it's just about being consistent with also learning that and just finishing one thing and then being, I guess, familiar with that and moving on to more advanced things.
Right. That's a very good point. And I have to go to Rabia and then I will come to Salman. Salman, there is a question from Ayman Yunus in the comments. If you are able to read that, we are going to take this question after I go to Rabia. So if you, in the meantime, if you are able to read that question from Ayman Yunus. And first of all, I will ask, this question is from Rabia. And many of the viewers are, I'm anticipating that this is going to be seen by some of the pharmacists. And as you know that your field is very much relevant to pharmacists, and I have this feeling that there are a few impediments in the mind of pharmacy students. First of all, like research in general in Pakistan is considered a very difficult area, not able to do thing. And then drug discovery and within research is something which is very, it's not considered as an easy area. So if an early career pharmacist wants to come into drug discovery, and what would you like to suggest to them?
I would suggest that start doing that when you are in a university setting, because then you have access to a lot of resources as a student. And then also you can communicate with the labs around the world who are doing research in drug discovery, and they will answer a lot of your questions. And most of the people are really nice, and they'll actually help you in whatever questions or problems you have. Of course, the best thing would be to join a lab that is working on drug discovery. And if I talk about myself, I do not have a background in pharmaceutical sciences or computing, but I mean, I was always interested in computers, and also I took some courses in bioinformatics and in molecular phylogenetics in the past, and also I did some projects during taking those courses. So, I mean, if somebody can just, I think if you can just learn from, like Osama said, the online courses from edX, etc., you can. There are tons of YouTube videos too. So I think start from there, try to educate yourself, and then reach out to people. And I think that will be the best thing.
Right. And before moving forward, I think I also say this thing to everybody who approaches me. And at the point that Panelists is referring to these things, the point when you use these resources and see the benefit of them, and the fact that they are free, and that I mean, if something comes to you for free, you tend to underestimate the importance of it. They're not they're very useful resources. That is something which we have to emphasize. And if you have to take these resources, like in the US and through a degree, you have to pay a huge amount of fee for the same level of skills which you have to develop. And with that, as I said, that I'm going to move forward to Salman with our next question, which is from Eman Yunus, and she's saying, did causality change according to race and ethnicity? Like, if some causal relationship is true, positive in Chinese race is also true in Kenya? Like, did race play any role?
Yeah, so it depends. What are you trying to explore here? But the definition of causality is that if you are claiming that this happens with everyone, then race should not matter. And if you are claiming that this particular phenomena happens with only one race, it should happen with that race, regardless he's in China or he's in Kenya. So that's the problem in establishing causality, because most of the time we don't know that it's because of that race or because of that country or because of that culture. Because Chinese can be in Kenya and have a hub there where they enjoy Chinese culture. So that's the problem in establishing causality. But if you are claiming causality, it should be true no matter what.
Okay, and so our next question is from here. Osama Shah is asking, are there any research facilities available in Pakistan as per Osama's genomic model? Osama?
So I can't say about many other institutes, but I did my undergrad from LUMS, and the genomic research is there, and but it's limited, I would say. Of course, given the setting, it's limited. And most of the important question in genomics that we can, I guess, answer using genomics in our country, apart from healthcare, is actually agriculture or plant genomics. That's a big topic, and like our government does give funding for those questions, which are related to like how to improve crop yields. So we do have people working in these plant genomic side at LUMS, at least. I'm not sure about the other places. And the other genomics, but I think still the sequencing part of it, like learning to use this big data, is limited.
Okay. Just a minute. I think there's some problem in my laptop. Just hold on for a second, okay? I'm going to adjust it.
I will add to that, to Osama's answer. At NUST, we have a few labs who work in genomics, both in plant sciences and animal sciences. And I remember one of the professors who was working in human genomics, he had funding to send samples to, I think, Germany and get them sequenced. So people, I mean, there are people who are doing some work, so you can do your research and find those labs and people. I mean, it's all of course limited, but still there is something happening.
Right. And the next question is Python or R. What is the use of these languages in cancer research?
Yeah. Okay. So it's not just the cancer research, the complexity of the genome. Like when you just talk about genomics, or even like anything which has a lot of information, you have, like, let's say, tweets from Twitter, right? Anything which has a lot of things, the way you like a lot of features you want to study, you would use some kind of computational method, and most of these require you to learn how to interact with computer, and that's why these languages like Python and R come in. So for genomics, as I mentioned, like there were a couple of things that I just put out, which was like sequencing of the sequence alignment. So like once you have, so let's say you do like DNA sequencing, and the genome is 3.2 billion base pair long. That's a lot of data. So thinking about in terms of like what you get out of the sequencing, you would need some methods to read it. And as the human eye cannot do that. So that's why you would use computers which could do pattern searching, right? Pattern matching, pattern searching. And that's what these languages can aid you in. So these methods are very time consuming, challenging. And that's why you use these languages to kind of like speed things up. When the first human genome was profiled, it took, I think it took a long time. I think it was the whole project was 10 years long. And most of that time went into the experimental part, which is the sequencing took a long time. And then, of course, the analysis also took a long time, but many of these problems have been solved. So what I mean is that the computational approaches have evolved a lot, and these help us speed up the analysis of genomic data.
Okay. And I think this will be our last question from the panel, from the comments. And then I have a few questions to conclude the discussion. And this question is, how can we apply bioinformatics in public health?
Okay. Yeah, I think some of the things that you can do with, so even if we don't think about the genomic data, let's forget about genomics for a while, because genomic data is very, again, the complexity is too much. But you have a lot of data like public health or clinical data available as well. So let's say the TCGA that Rabia also mentioned, that is the Cancer Genome Atlas data. So not only have they done the RNA, DNA sequencing and many other things, they also have clinical data. So let's say you want to find, okay, what is the survival of a particular patient if they have high expression of a particular gene or mutation in a particular gene. And even at a more basic level, you can think like, how does age relate with survival? Let's say, how does a particular treatment relate with survival? So I think these things are more at the basic level more important when you do like public health research. And in Pakistan, you would want to know like, so people could start generating data sets. I'm not aware of how many proper data sets are out there, but the idea would be to keep a tag on patients. Let's say you get a patient with a particular cancer or even a particular disease. What treatments were given? How does this, and when did the person, I guess, have an outcome? Let's say that could be death, or that could be recurrence of a disease and something like that. So all these data sets could be used in the public health domain and analyze using bioinformatics approaches to better understand how we can shape our own healthcare system in terms of like management and all these things. Yeah, so if you want to like use bioinformatics in public health, I would say you can again do it in these clinical using these clinical data sets where you have features like treatments and what outcome was achieved using these treatments for different diseases, and you can go towards more advanced side, which is the genomics as well. So yeah, both of these could be used.
Okay, my last question is from Salman. As Rabia and Osama mentioned, a lot of online sources. So because a lot of people who are watching this are public health professionals or epidemiologists. So my question is that, like, as Rabia and Osama said, that there are free online courses for Python or R. Are there any online resources for econometrics? And if I'm an epidemiologist and I want to diversify my skill set by learning econometrics, so would you? Are there any resources, and would you recommend some of them?
Yeah, there are many resources. Again, the Coursera and edX are very good sources, but I think the Stanford has a online. It's not an online course, but there are lectures available on causality in the economics department. I think that was very helpful when I was studying this.
Okay, so the one theme is coming out from this particular discussion. Everybody is referring to, recommending Coursera, edX, YouTube, and like some university online courseware. So these are the things which should be seen on a regular basis. With that note, I'm going to thank our panelists. Rabia, Khurshid, thanks a lot for coming to us. We know that you had some health problems and you stayed with us until the end, and it's not easy. Thanks a lot. Osama Shiraz, thanks a lot for taking your time from your busy schedule. Salman, once again, like you also, thanks a lot to you as well. And thanks to the viewers who have been staying with us until the end and who helped generate an engaging discussion by asking questions and taking part in this discussion. So with that, we will keep on posting or organizing similar events in the future. If you want to give us any feedback, I have 3 specific questions for the viewers. What was the good thing about this session? What was something which you want us to improve, or any generic suggestion which you want to give, which will help us improve the quality of these sessions in the future? With that note, I will say take care for time being. Bye and Allah Hafiz.
Allah Hafiz. Bye-bye. Thanks.